So here you are, lying in the dark, and your body is doing about 10,000 things right now that you didn't ask it to do. Your heart is beating. Your lungs are filling and emptying. Somewhere deep in your gut, enzymes are breaking apart whatever you had for dinner, pulling it into molecules small enough to slip through the wall of your intestine and into your blood. Your immune system is quietly patrolling, checking credentials, making sure nothing got in that shouldn't be there. And your brain is doing the most extraordinary thing of all, which is listening to my voice and turning vibrations in the air into language, into meaning, into something that feels like understanding. You didn't have to think about any of that. It's just happening. All of it, right now, in the dark. And here's what I want to do tonight. I want to walk you through how all of it works. Not quickly, not in some rushed overview, but slowly, carefully, the way you'd explain something to someone you really liked at the end of a long evening. Because the human body is, without exaggeration, the most complex system anyone has ever tried to understand. More complex than any bridge, any spacecraft, any computer network. And yet it builds itself. It repairs itself. It runs on sandwiches and sleep. That's genuinely astonishing when you stop to think about it. But let's not start with the body as it is right now. Let's start with the people who first tried to figure it out. Because for most of human history, the inside of a living body was a complete mystery. You could see the outside, skin, eyes, hair, the shape of muscles under the surface. You could see what came out of the body, blood from a wound, breath in cold air. But what was actually going on in there? That was guesswork. Beautiful, elaborate, sometimes wildly wrong guesswork. Think about ancient Egypt, around 1600 BCE. We have a document called the Edwin Smith Papyrus. It's named after the American collector who bought it in Luxor in 1862. But the text itself is almost 3,500 years old. And what's remarkable about it is how practical it is. It describes 48 surgical cases, starting from the head and working down, and for each one it gives a diagnosis, a treatment, and a prognosis. It talks about the brain. And here's the thing. The Egyptian who wrote this actually noticed that injuries to one side of the brain could cause problems on the opposite side of the body. That's real observation. That's someone paying attention at the bedside and writing down what they saw. But even with that level of careful observation, the Egyptians still believed the heart was the seat of intelligence. The brain, they thought it was basically packing material. When they mummified someone, they'd carefully preserve the heart, the liver, the lungs, the intestines, but the brain they'd pull out through the nose with a hook and throw it away. Just discard it. The organ that makes you who you are, and they tossed it. And you can't really blame them, right? If you open up a skull and look at a brain, it doesn't look like it's doing anything. It's this soft, wrinkled, grayish mass. It doesn't beat like a heart. It doesn't expand like a lung. It just sits there. Meanwhile, the heart is dramatic. You can feel it pounding in your chest when you're scared. You can feel it race when you're in love. Of course, they thought the heart was where thinking happened. It took thousands of years to sort that out. Now, the person who really started pushing anatomy forward in the ancient Western world was a Greek physician named Hippocrates, born around 460 BCE on the island of Kos. You've probably heard of him, the Hippocratic Oath, all of that. But what made Hippocrates genuinely revolutionary wasn't any single discovery. It was an idea. He proposed that disease had natural causes, not divine punishment, not evil spirits, not curses, natural, physical causes that you could observe, reason about, and potentially treat. That sounds obvious now. But in a world where illness was routinely attributed to angry gods, it was a seismic shift in thinking. Hippocrates and his followers developed the theory of the four humors: blood, phlegm, yellow bile, and black bile. The idea was that health depended on these four fluids being in balance. Too much blood made you hot and wet. Too much black bile made you cold and dry. If you were sick, something was out of balance, and the physician's job was to restore equilibrium. Now, this was wrong. Completely wrong, as it turned out. But it was wrong in an incredibly productive way, because it gave physicians a framework for thinking systematically about the body. It encouraged them to observe symptoms carefully, to look for patterns, to consider diet and environment and lifestyle. The humoral theory dominated Western medicine for nearly 2,000 years, which tells you something about how powerful a coherent framework can be, even when the underlying assumptions are off. But let's jump forward a few hundred years to someone who took anatomy much further, a physician named Galen, born around 129 CE in Pergamon, which is in modern-day Turkey. Galen is one of the most influential figures in the entire history of medicine, and also one of the most complicated. He was brilliant, arrogant, prolific, and wrong about a surprising number of things, but wrong in ways that shaped medicine for over a millennium. Galen was the personal physician to Roman emperors. He treated gladiators early in his career, which gave him an unusual window into the human body. Literally. Gladiatorial wounds exposed muscles, tendons, and organs in ways that normal medical practice never would. He called those wounds windows into the body, and he learned from them. But here's the problem. Roman law and custom prohibited the dissection of human corpses. So when Galen wanted to study internal anatomy in detail, he had to use animals, pigs, monkeys, goats, dogs. He dissected Barbary macaques and assumed their anatomy was essentially identical to human anatomy. Sometimes it was close. Sometimes it really wasn't. For example, Galen described the rete mirabile, a network of blood vessels at the base of the brain. It's a real structure, in ungulates, in cows and sheep and pigs. But humans don't have one. Galen said we did, because the monkeys he was dissecting had something sort of like it, and he extrapolated. And because Galen's authority was so enormous, because he wrote so much, so confidently, and because the Roman and later the medieval world treated his texts almost like scripture, that error persisted for over 1,000 years. Anatomists would look at a human body, not find the rete mirabile, and assume they'd made a mistake. Not Galen. Them. Think about what that means. For century after century, the authority of a text overrode the evidence of direct observation. That's a very human tendency, and it didn't just affect medicine. But in anatomy, the consequences were real. Galen's model of the circulatory system, for instance, held that blood was produced in the liver, flowed outward through the veins, and was consumed by the body's tissues like fuel. It didn't circulate. It was manufactured and used up continuously, like logs on a fire. The heart, in Galen's view, wasn't really a pump. It was more like a furnace that heated the blood and mixed it with a vital spirit drawn in from the lungs, and blood passed from the right side of the heart to the left through tiny, invisible pores in the septum, the thick wall of muscle between the heart's chambers. Those pores don't exist. The septum is solid. But Galen said they were there, so for 1,500 years, anatomists said they were there too. Now, here's where it starts to get really interesting. The person who finally cracked open Galen's authority who looked at the actual body and said, "No, this is what's really here," was a young Belgian anatomist named Andreas Vesalius. And his story is one of those moments in the history of science where one person's stubbornness changes everything. Vesalius was born in Brussels in 1514 into a family of physicians and pharmacists. He was apparently obsessed with anatomy from a very young age. There are stories of him dissecting mice and stray cats as a child, which is either precocious or alarming depending on your perspective. He studied at the University of Paris, where anatomy was taught in the traditional way. A professor would sit in an elevated chair, reading aloud from Galen's texts, while a barber surgeon down below did the actual cutting. The professor never touched the body. The students watched from a distance. And what was being read from the book was treated as more authoritative than what was lying on the table. Vesalius found this maddening. He started doing his own dissections, sometimes obtaining bodies by questionable means. There's a well-known story about him stealing a corpse from a roadside gibbet outside the city walls of Leuven, carrying the bones home in pieces over multiple trips. He was driven, and he was meticulous, and he was increasingly convinced that Galen had made errors, not small errors, fundamental errors about the structure of the human body. In 1543, when he was just 28 years old, Vesalius published De Humani Corporis Fabrica, On the Fabric of the Human Body. It's one of the most important books ever printed. 7 volumes, over 700 pages, with detailed illustrations that were probably produced in the workshop of Titian, the great Venetian painter. The images are stunning. Flayed figures standing in landscapes, posed like classical sculptures, their muscles and tendons rendered with an artist's eye and an anatomist's precision. But what made the Fabrica revolutionary wasn't the art. It was the insistence on observation over authority. Vesalius corrected over 200 of Galen's errors. He showed that the human jaw is one bone, not 2 as Galen had described from monkeys. He showed that the septum of the heart is solid, with no pores. He showed that the human liver has no distinct lobes the way an animal's does, and the medical establishment was furious. His former teacher, Jacobus Silvius, called him a madman, a slanderer of Galen. Silvius actually argued that the human body must have changed since Galen's time, that tight clothing had somehow altered human anatomy over the centuries. That was easier to believe, apparently, than the possibility that Galen had been wrong. But Vesalius was right, and slowly, painfully, the world of anatomy began to shift from a tradition based on ancient texts to one based on direct observation. It's hard to overstate how important that shift was, because everything we know about how your body works, everything I'm going to tell you tonight, traces back to that fundamental commitment. Look at the thing. Don't just read about it. Look at it. Measure it. Cut it open if you have to. See what's actually there. And once people started really looking, the discoveries came faster. Within a century of Vesalius, the whole understanding of how blood moves through your body would be overturned. A quiet English physician would figure out something that Galen had gotten completely backward, and it would change medicine forever. His name was William Harvey, and what he discovered was so simple, so elegant, and so obviously correct that you have to wonder how anyone ever believed anything else. But to understand what Harvey figured out, you first have to understand what people thought was happening inside your chest. This warm, dark, rhythmically thumping space right behind your ribs, and why the old explanation, wrong as it was, made a kind of intuitive sense that was very hard to let go of. Because the heart, when you really start to look at what it does and how it does it, turns out to be one of the most extraordinary pumps ever devised. And I mean that literally. It's a pump. That's what it is. But for most of human history, people didn't think of it that way at all. Under Galen's model, which remember had dominated medicine for nearly 1,500 years by the time Harvey came along, the heart wasn't really a pump. It was more like a furnace, a source of heat. The idea was that blood was manufactured in the liver from the food you ate. Your liver would somehow cook your digested food into dark, venous blood, and then that blood would flow outward through your veins to every part of your body, where it would be consumed, used up, like fuel. Your tissues would just absorb it, the way a sponge absorbs water, and that was that. Meanwhile, a small amount of blood would seep through tiny invisible pores in the wall between the left and right sides of the heart, the septum, and mix with air from your lungs, becoming a different, brighter kind of blood called arterial blood, which carried what Galen called vital spirit. This arterial blood would then flow out through the arteries to give your body life and warmth, and it too would be consumed at the periphery. Gone. Used up. Your liver would just keep making more. So in Galen's world, blood was a consumable. It was constantly being produced, constantly being used, and it flowed in one direction, outward, like water from a spring, spreading out and soaking into the ground. There was no return trip, no circulation, no loop. And honestly, if you'd never done the math, if you'd never sat down and really thought carefully about the quantities involved, that explanation kind of works. It feels right. You eat food, your body turns it into blood, the blood nourishes your tissues. Simple, elegant, even. The problem is that it's completely wrong. William Harvey was born in Folkestone, England, in 1578. He studied at Cambridge, then traveled to Padua, the same University of Padua where Vesalius had done his revolutionary dissections decades earlier. Padua was the place to study anatomy in those days, and Harvey's teacher there was a man named Hieronymus Fabricius, who had made a curious discovery about veins. Fabricius had noticed that veins contained tiny flap-like structures inside them, little one-way valves. He described them. He drew them. But he didn't quite understand what they were for. He thought they might slow the flow of blood to prevent it from pooling in the extremities. He was close, but he missed the real significance. Harvey would figure it out. After returning to England, Harvey became a physician at St. Bartholomew's Hospital in London and was eventually appointed physician to King James I and later to Charles I. He had access to patients, to cadavers, and crucially to a wide variety of living animals for dissection. And he was meticulous, patient, the kind of person who would sit and watch a beating heart for hours, trying to understand the sequence of its movements, the timing of each contraction, the direction of blood flow through each chamber. He did this with cold-blooded animals, fish, frogs, snakes, because their hearts beat slowly enough to observe. A human heart beats roughly 70 times a minute. A frog's heart might beat only 20 or 30 times. You can actually watch each phase of the contraction unfold. And here's what Harvey noticed, just by watching carefully. When the heart contracts, when it squeezes, it gets smaller and harder, like a flexed muscle. And when it does that, blood is pushed out of it, not drawn in. Pushed out. That might seem obvious to you now, but at the time, people thought the heart's active phase was when it expanded, when it dilated, that it was sucking blood in, the way a bellows draws in air. Harvey showed it was the opposite. The heart is a muscle, and like any muscle, it does its work when it contracts. The expansion is the resting phase. The contraction is the working phase. The heart squeezes blood out into the arteries with each beat. Okay, so far so good. But here's where Harvey's genius really shows. He did something that nobody in medicine had really done before, at least not with this problem. He did the math. He estimated the capacity of the left ventricle, the main pumping chamber of the heart, at roughly 2 ounces of blood per beat. That's a conservative estimate. The real number is a bit higher, but Harvey deliberately chose low numbers to make his argument harder to dismiss. 2 ounces per beat, 72 beats per minute. That's about 144 ounces per minute. In an hour, that's over 500 pounds of blood. In a day, thousands upon thousands of pounds. Now think about what that means under Galen's model. If blood is being manufactured fresh by the liver and consumed at the periphery, your liver would need to produce thousands of pounds of blood every single day from the food you eat, and your body would need to consume that same amount. That's obviously impossible. You don't eat thousands of pounds of food a day. You eat a few pounds at most. The numbers simply don't work. The math breaks the entire model. The only explanation that makes sense, the only way to account for that volume, is that the same blood is being used over and over again. It's not being consumed. It's circulating. It goes out through the arteries, reaches the tissues, and then comes back through the veins to the heart, where it's pumped out again. A closed loop, a circuit. That's what the word circulation means. It literally comes from the Latin word for circle, and those little valves that Fabricius had found in the veins, they suddenly made perfect sense. They're one-way valves. They allow blood to flow toward the heart, but not away from it. They're there to ensure the return trip works, especially in your legs, where blood has to fight gravity to get back up to your chest. Every time your leg muscles contract, when you walk, when you shift your weight, they squeeze the veins and push blood upward, and the valves prevent it from falling back down. It's like a series of locks in a canal, lifting the blood step by step back to the heart. Harvey published his findings in 1628 in a slim, unassuming book called Exercitatio Anatomica de Motu Cordis et Sanguinis in Animalibus, an anatomical exercise on the motion of the heart and blood in animals. It's usually just called De Motu Cordis, and it's one of the most important books in the history of medicine. Not because it's long or elaborate, it's actually quite short and plainly written, but because it replaced a 1,500-year-old wrong answer with the right one, and it did so using evidence, observation, and quantitative reasoning. Harvey didn't just say, "I think blood circulates." He showed it. He measured it. He proved it. Now, there was one piece of the puzzle Harvey couldn't solve. He knew blood went from the arteries to the veins somehow, but he couldn't see how. The connection between the smallest arteries and the smallest veins was invisible to the naked eye. He hypothesized that tiny passages must exist, but he couldn't prove it. That final piece wouldn't come until 1661, when an Italian physician named Marcello Malpighi turned one of the newly invented microscopes on the lung tissue of a frog and saw, for the first time, capillaries. The tiniest blood vessels in your body, so thin that red blood cells have to pass through them in single file. Malpighi saw blood flowing from the arterial side through these impossibly delicate vessels and into the venous side. The circuit was complete, and that's worth sitting with for a moment. Your body contains somewhere around 60,000 miles of blood vessels. 60,000 miles. That's enough to wrap around the earth more than twice. Most of that length is capillaries, these microscopic tubes woven into virtually every tissue you have. They're where the real work happens. The arteries are just the highway getting blood to the neighborhood. The capillaries are the local streets where deliveries are actually made. Oxygen dropped off, carbon dioxide picked up, nutrients exchanged, waste collected. Every living cell in your body is within a hair's width of a capillary. That's not an accident. That's engineering. And the heart driving this whole system. It beats about 100,000 times a day, roughly 35 million times a year. Over an average lifetime, that's somewhere around 2.5 to 3 billion beats, without stopping, without maintenance, without a single conscious instruction from you. Right now, as you're lying there, your heart is beating. You don't have to think about it. You don't have to remember to do it. It just goes. Here's what's really beautiful about how it works. The heart is actually two pumps in one, sitting side by side, wrapped in the same muscular wall. The right side receives oxygen-poor blood from your body, blood that's already made its delivery run, dropped off its oxygen, picked up carbon dioxide, and pumps it to your lungs, just to your lungs. A short trip. The blood passes through the capillaries in your lungs, dumps its carbon dioxide, picks up fresh oxygen, and returns to the left side of the heart. Then, the left side, which is noticeably thicker and more muscular because it has a harder job, pumps that freshly oxygenated blood out to your entire body. Every organ, every muscle, every fingertip and earlobe, and the tip of your nose. 2 pumps, 2 circuits, working in perfect synchrony, every single beat. The coordination of this is remarkable. Your heart has its own built-in electrical system, a cluster of specialized cells called the sinoatrial node, tucked into the wall of the right atrium, which fires an electrical impulse roughly once a second. That impulse spreads across both atria, the upper chambers, causing them to contract and push blood down into the ventricles. Then the signal hits another node, the atrioventricular node, which delays it for just a fraction of a second, about 1/10 of a second, to give the ventricles time to fill completely before they contract. Then, the signal races down a bundle of specialized fibers through the septum and fans out across both ventricles, triggering that powerful squeeze that sends blood surging into the arteries. The whole sequence, atria contract, brief pause, ventricles contract, takes less than a second, and it repeats and repeats and repeats. The sinoatrial node is sometimes called the heart's natural pacemaker, and it doesn't need any input from your brain to keep firing. If you took a heart completely out of a body and kept it supplied with oxygenated, nutrient-rich fluid, it would keep beating on its own. It's self-sustaining. Your nervous system can speed it up or slow it down. Adrenaline makes it race, rest lets it slow. But the basic rhythm is intrinsic. It's built into the tissue itself, and the sound of your heartbeat, that familiar lub-dub you've heard through a stethoscope or felt in your own chest. Those aren't the sounds of the muscle contracting. They're the sounds of valves closing. The first sound, the lub, is the mitral and tricuspid valves snapping shut as the ventricles begin to contract, preventing blood from flowing backward into the atria. The second sound, the dub, is the aortic and pulmonary valves closing as the ventricles relax, preventing blood from flowing backward out of the arteries. It's the sound of doors slamming shut at exactly the right moment, ensuring that every drop of blood goes in the right direction. When a doctor listens to your heart with a stethoscope, what they're really listening for is any deviation from that clean lub-dub. A murmur, a whooshing or swishing sound, might mean one of those valves isn't closing properly, or has become narrowed, or has a small leak. The valves are astonishingly thin and delicate, but they open and close 3 billion times in a lifetime. Think about any mechanical valve you've ever seen, a faucet, a check valve in plumbing, and imagine it cycling 3 billion times without replacement. The engineering tolerance is extraordinary. But the heart, magnificent as it is really just the pump. The delivery system, the vasculature, is where things get even more interesting, because your blood vessels aren't just passive pipes. They're active, responsive, constantly adjusting. The walls of your arteries contain smooth muscle that can contract or relax, narrowing or widening the vessel to control blood flow and pressure. When you're exercising, the arteries feeding your muscles dilate to deliver more oxygen. When you're cold, the vessels near your skin constrict to conserve heat, which is why your fingers turn pale in winter. When you're digesting a big meal, blood flow to your gut increases, which is part of why you feel sleepy after eating. There's a bit less blood available for everything else. This constant, dynamic reallocation of blood flow is one of the most sophisticated control systems in your body, and it's happening right now, beneath your awareness, adjusting to your current state, lying still, breathing slowly, drifting towards sleep. Your heart rate is probably dropping. Your blood pressure is easing down. The vessels in your core are gently dilating, while the ones in your extremities might be doing the opposite. Your body is quietly, expertly managing a fluid dynamics problem that would challenge any engineer, and it's doing it without you having to think about a single thing. But blood itself, the fluid flowing through all those miles of vessels, what actually is it? What's in it? Because when you see blood, it just looks like a red liquid. Simple. Uniform. But if you could shrink yourself down and float inside a blood vessel, what you'd find is something astonishingly crowded. It's not a uniform liquid at all. It's more like a river teeming with life, a thick, busy suspension of cells and proteins and dissolved gases, all tumbling along together through these narrow channels, and the sheer number of things in there is staggering. Let's start with the most obvious residents: red blood cells, erythrocytes if you want the formal name. These are the ones that give blood its color, and they are by far the most abundant cells in your bloodstream. In a single drop of blood, just one tiny drop balanced on the tip of your finger, there are roughly 5 million red blood cells. 5 million in a drop. And you've got about 5 liters of blood in your body, so the total count is somewhere around 25 trillion red blood cells circulating through you right now. That number is so large it's almost meaningless. But try this. If you could line them up edge to edge, they'd wrap around the earth multiple times. And every single one of them has one job: carrying oxygen. Here's what's beautiful about their design. A red blood cell is shaped like a biconcave disc. Imagine a donut, but instead of a hole in the middle, it's just pinched inward on both sides. It's a shallow, flexible dimple. And that shape isn't random. It maximizes the surface area relative to the cell's volume, which means more membrane is exposed to surrounding fluid at any given moment, making gas exchange faster and more efficient. But there's another reason for that shape. Red blood cells need to squeeze through capillaries that are narrower than the cells themselves. Some capillaries are only 3 or 4 micrometers wide, and a red blood cell is about 7 or 8 micrometers across. So the cell has to fold, bend, and deform to pass through. That biconcave shape, combined with a remarkably flexible membrane, lets it do exactly that. It sort of folds in on itself like a taco, slips through the tight space, then springs back to its original shape on the other side. It does this thousands of times during its lifespan, which is about 120 days. After that, It's worn out, and your spleen, which acts as a kind of quality control station, filters it out and breaks it down so the components can be recycled. Your body is producing roughly 2 million new red blood cells every second to replace the ones being retired. 2 million per second, just to maintain a steady supply. Now, the magic molecule inside each red blood cell is hemoglobin. This is the protein that actually grabs onto oxygen. Each hemoglobin molecule contains 4 iron atoms, and each iron atom can bind 1 molecule of oxygen. So each hemoglobin carries 4 oxygen molecules, and each red blood cell contains about 270 million hemoglobin molecules. The math gets absurd, but the point is that your blood has an enormous oxygen carrying capacity, far more than if oxygen were simply dissolved in the liquid portion of blood. In fact, if you relied only on dissolved oxygen without hemoglobin, you'd need something like 80 liters of blood to deliver enough oxygen to your tissues. Hemoglobin increases the oxygen carrying capacity by about 70 times. It's the difference between trying to move a pile of bricks by carrying them one at a time versus loading them onto a fleet of trucks. And here's what's really clever about hemoglobin. It doesn't just grab oxygen and hold on. It has this property called cooperative binding. When the first oxygen molecule attaches to one of the 4 binding sites, it changes the shape of the hemoglobin molecule slightly, making it easier for the second oxygen to attach. And the second makes the third easier, and so on. This means that in the lungs, where oxygen concentration is high, hemoglobin loads up very efficiently. It practically vacuums up oxygen. But then when the red blood cell travels to a tissue that's using oxygen, where the concentration is lower and the environment is slightly more acidic from carbon dioxide and metabolic waste, the hemoglobin's grip loosens. It releases oxygen more readily in exactly the places that need it most. This elegant little feedback loop is described by something called the oxygen-hemoglobin dissociation curve, and it's one of those things in physiology that just makes you sit back and appreciate how well-tuned the system is. The molecule itself is smart about where to hold on and where to let go. The iron in hemoglobin, by the way, is why blood is red. When oxygen binds to the iron, it changes the way the molecule absorbs light, giving oxygenated blood that bright scarlet color. Deoxygenated blood that's already delivered its oxygen and is heading back to the lungs, is a darker, more muted red. Not blue, despite what you might have been told in school. The veins look blue through your skin because of the way light penetrates and scatters through tissue, but the blood inside them is always some shade of red. So that's the red blood cells, the oxygen couriers. But they're not the only cells in the river. Floating alongside them, much less numerous but arguably just as important, are the white blood cells, leukocytes. If red blood cells are the delivery trucks, white blood cells are the security force. They're the immune system's frontline agents, and they come in several varieties, each with a different specialization. Neutrophils are the most common, making up about 60 to 70% of your white blood cells, and they're the first responders. When bacteria invade through a cut in your skin, neutrophils are the ones that arrive within minutes, engulfing and destroying the intruders through a process called phagocytosis, literally eating them. A single neutrophil can consume and digest about 5 to 20 bacteria before it dies from the effort. The pus you see in an infected wound is largely a collection of dead neutrophils that gave their lives in battle. There's something almost heroic about that, in a cellular kind of way. Then there are lymphocytes, the B cells and T cells you've probably heard of, which handle the more sophisticated, targeted immune responses. B cells produce antibodies, those Y-shaped proteins that lock onto specific invaders like a key fitting a lock. T cells come in several flavors: helper T cells that coordinate the immune response, and killer T cells that destroy your own cells when they've been infected by viruses or have turned cancerous. The specificity of this system is remarkable. Your body can produce antibodies against virtually any foreign molecule it encounters, even synthetic chemicals that have never existed in nature. It does this through a process of genetic recombination in developing lymphocytes that generates an almost infinite diversity of antibody shapes. It's like having a locksmith who can cut a key for any lock in the universe, even ones that haven't been built yet. And then there are the platelets, technically not cells at all, but fragments of much larger cells called megakaryocytes that live in your bone marrow. Platelets are tiny, irregularly shaped, and they're the emergency repair crew. When a blood vessel is damaged, the exposed collagen fibers in the vessel wall trigger platelets to activate. They change shape, becoming sticky and spiky, and they clump together at the site of injury, forming a plug. Then a cascade of clotting factors, a chain reaction involving more than a dozen different proteins in the blood, amplifies the response, ultimately producing fibrin, a tough, thread-like protein that weaves through the platelet plug like reinforcing mesh, creating a stable clot. This clotting cascade is one of the most intricate biochemical chain reactions in the body, and it has to be precisely balanced. Too little clotting, and you bleed uncontrollably from minor injuries. Too much, and you form clots inside intact blood vessels, which can block blood flow and cause strokes or heart attacks. The body walks this tightrope constantly. Now, all of these cells, the red blood cells, the white blood cells, the platelets, they're all suspended in a pale, straw-colored liquid called plasma, which makes up about 55% of your blood's volume. Plasma is mostly water, about 92%, but dissolved in that water is an extraordinary pharmacy of substances. Proteins like albumin, which helps maintain the osmotic pressure that keeps fluid from leaking out of your blood vessels. Immunoglobulins, antibodies, patrolling for threats. Clotting factors, standing by. Hormones being ferried from the glands that produce them to the distant tissues they're meant to influence. Glucose, the primary fuel for your cells. Electrolytes, sodium, potassium, calcium, chloride, in carefully maintained concentrations that your kidneys monitor and adjust continuously. Waste products like urea and creatinine being carried to the kidneys for disposal. Dissolved carbon dioxide, heading back to the lungs to be exhaled. It's an incredibly busy solution. And this brings us to something worth lingering on: the kidneys. Because if the heart is the pump and the blood vessels are the pipes, the kidneys are the filtration plant. And what they do is, frankly, one of the most underappreciated feats of engineering in your entire body. Each kidney is only about the size of your fist, maybe 4 or 5 inches long. And yet together, they filter your entire blood supply roughly 40 times a day. That's about 180 liters of fluid filtered every 24 hours. Obviously, you don't urinate 180 liters a day. You'd be in serious trouble if you did, because the kidneys reabsorb about 99% of that filtered fluid, sending it back into the blood. Only about 1 to 2 liters ends up as urine. The kidneys are selective, precise, and incredibly efficient at deciding what stays and what goes. The functional unit of the kidney is called the nephron, and each kidney contains about 1 million of them. A nephron is essentially a tiny tube with a very specific architecture. At one end is a structure called the glomerulus, a tight knot of capillaries enclosed in a cup-shaped capsule called Bowman's capsule. Blood enters the glomerulus under pressure, and that pressure forces water and small dissolved molecules through the capillary walls and into the capsule, like pressing fluid through a coffee filter. Large molecules like proteins and blood cells are too big to pass through, so they stay in the blood. What passes through, the filtrate, then travels down a long, winding tubule, and this is where the real sophistication happens. Different segments of the tubule are specialized for reabsorbing different substances. Glucose gets pulled back in. Amino acids get reclaimed. Sodium and chloride are actively pumped back into the blood in carefully regulated amounts. Water follows by osmosis, drawn by the concentration gradients the tubule cells create. By the time the filtrate reaches the collecting duct at the end of the nephron, it's been transformed from a dilute, everything-included filtrate into concentrated urine, containing mostly waste products. The kidneys also play a critical role in regulating blood pressure. They can adjust how much sodium and water they retain, which directly affects blood volume and therefore pressure. They produce a hormone called erythropoietin that signals the bone marrow to make more red blood cells when oxygen levels drop. They help regulate the pH of your blood, keeping it in that incredibly narrow range between about 7.35 and 7.45, because even slight deviations outside that range can denature proteins and shut down enzymes. The kidneys are quietly performing all of these functions right now as you lie there, and they've been doing it every moment of your life since before you were born. But the kidneys are really just one part of a larger theme, which is how your body maintains its internal environment, how it keeps everything stable despite a constantly changing external world. Physiologists have a word for this, and it's one of the most important concepts in all of biology. It was coined in 1926 by a Harvard physiologist named Walter Cannon, building on ideas that the great French physiologist Claude Bernard had articulated decades earlier. Bernard had observed that the internal environment of the body, the milieu intérieur he called it, remains remarkably constant even as external conditions fluctuate. Cannon gave this principle a name that stuck: homeostasis. And once you understand homeostasis, you start to see it everywhere in the body, governing temperature, blood sugar, hydration, pH, oxygen levels, hormone concentrations, essentially every measurable parameter of your internal state. The mechanisms that maintain homeostasis are varied and complex, but they almost all share a common logic, a design pattern that engineers would immediately recognize because it's the same principle that governs a thermostat in your house, a cruise control system in your car, or a governor on a steam engine. It's called negative feedback, and the way it works in your body is beautifully simple. Your body sets a target value for something, let's say temperature, and then it monitors the actual value continuously. When the actual value drifts away from the target, the body activates a response that pushes it back. And here's the key part. As the value returns toward the target, the response itself diminishes. The correction is self-limiting. That's what makes it negative feedback. The output of the system feeds back to reduce the input that triggered it. It's elegant and it's everywhere. Think about your body temperature for a moment. You're lying there right now, and your core temperature is hovering somewhere around 37 degrees Celsius, which is about 98.6 Fahrenheit, give or take a degree depending on the time of day and your own personal baseline. That number isn't arbitrary. It's the temperature at which your enzymes, those protein catalysts that drive virtually every chemical reaction in your body, work most efficiently. Too cold, and reactions slow to a crawl. Too hot, and the proteins start to unfold, losing their shape and their function. So your body defends that temperature fiercely, and the command center for this defense sits in a tiny region of your brain called the hypothalamus. The hypothalamus is remarkable. It's only about the size of an almond, tucked underneath the larger brain structures, and yet it acts as the master thermostat for your entire body. It receives temperature information from two sources: thermoreceptors in your skin that detect the temperature of your environment, and thermoreceptors in the blood flowing through the hypothalamus itself, which tell it the core temperature directly. When you step outside on a cold winter night, the skin receptors fire first. They're the early warning system, telling your hypothalamus that heat loss is about to become a problem before your core temperature has actually dropped. And then, the hypothalamus orchestrates a response. It triggers shivering, which is just rapid, involuntary muscle contractions that generate heat as a byproduct. It causes vasoconstriction, the narrowing of blood vessels near your skin surface, which reduces blood flow to the periphery and keeps warm blood closer to your vital organs. It can even trigger hormonal responses that ramp up your metabolic rate, burning more fuel to produce more heat. And as your core temperature returns to its set point, all of those responses dial back. The shivering stops. The blood vessels relax. You reach equilibrium again. That's negative feedback in action. Now flip it around. You're exercising on a hot day. Your muscles are generating enormous amounts of heat. Remember, muscles are only about 20 to 25% efficient at converting chemical energy into mechanical work, so the rest becomes thermal energy. Your core temperature starts to rise. The hypothalamus detects this and triggers the opposite set of responses. Vasodilation. The blood vessels near your skin open wide, flooding the surface with warm blood so heat can radiate away. You start sweating, and as that moisture evaporates from your skin, it carries heat with it. This is remarkably effective. Evaporation of sweat can dissipate more than 600 watts of heat energy under ideal conditions, which is more than some space heaters produce. And again, as your temperature drops back toward the set point, the sweating slows and the vasodilation eases. Here's what's really clever about this system. It's not just one feedback loop. It's dozens of them, layered and interconnected, operating at different speeds and different scales. Some are fast, like the neural reflexes that trigger shivering within seconds. Some are slow, like hormonal adjustments to your metabolic rate that unfold over hours or days. And they interact with each other in ways that create a robustness you wouldn't get from any single mechanism alone. If one system fails or is overwhelmed, others compensate. It's redundancy built into biology, and it's the same design philosophy that engineers use in critical systems. Aircraft have backup hydraulics, power grids have redundant transmission lines, and your body has multiple overlapping pathways to maintain every important variable. But here's the thing about homeostasis that people sometimes miss. It's not about keeping everything perfectly constant. It's about keeping everything within an acceptable range despite constant disturbance. Your body temperature actually fluctuates throughout the day. It's typically lowest in the early morning, around 4 or 5 a.m., and highest in the late afternoon. Your blood pressure changes with every heartbeat, every change in posture, every emotional reaction. Your blood glucose rises after a meal and falls between meals. The system isn't rigid. It's dynamic. It's constantly adjusting, constantly correcting. Like a tightrope walker who never stops moving, but never falls. And that brings us to one of the most consequential homeostatic systems in your body, one that affects billions of people, and one that, when it breaks down, causes one of the most common diseases in the modern world. Let's talk about blood sugar regulation, because the story of how we came to understand it is one of the great dramas in the history of medicine. Your body runs on glucose. Every cell in your body can use glucose as fuel, and your brain is particularly dependent on it. The brain accounts for only about 2% of your body weight, but consumes roughly 20% of your glucose supply. So maintaining a steady supply of glucose in your blood is critical. Too little, and your brain starts to malfunction. You get confused, shaky, you can lose consciousness. Too much, and the excess glucose starts to damage blood vessels, nerves, kidneys, eyes. The target range is roughly 70 to 100 milligrams per deciliter of blood when you haven't eaten recently. Your body works hard to keep it there. The key player in this system is a hormone called insulin, and the story of its discovery is extraordinary. By the late 1800s, physicians knew that there was a disease, diabetes mellitus, in which patients had chronically elevated blood sugar, passed sugar in their urine, lost weight despite eating voraciously, and eventually died. They knew it had something to do with the pancreas, because in 1889, two researchers in Strasbourg, Oskar Minkowski and Joseph von Mering, had removed the pancreas from a dog and noticed that the animal developed severe diabetes almost immediately. Flies swarmed the dog's urine because it was full of sugar. That was the clue. Something the pancreas produced was essential for controlling blood sugar. But what was it? The pancreas is mostly an organ that produces digestive enzymes, powerful proteins that break down food in your small intestine. Scattered throughout this digestive tissue, however, are tiny clusters of cells that look different under a microscope. A German medical student named Paul Langerhans had described them in 1869 in his doctoral thesis, and they became known as the islets of Langerhans. Researchers suspected that these islets produced something, some internal secretion, that regulated blood sugar. But every time anyone tried to extract it from the pancreas, the digestive enzymes destroyed it before they could isolate it. It was maddening. For 30 years, researchers tried and failed. And then, in the fall of 1920, a young Canadian surgeon named Frederick Banting had an idea. Banting was not a researcher. He was a general practitioner in London, Ontario, struggling to build a practice, teaching part-time at Western University to make ends meet. While preparing a lecture on the pancreas, he read a paper about how tying off the pancreatic duct caused the digestive enzyme-producing tissue to wither away, while the islets of Langerhans survived. And in the middle of the night, the story goes that it was around 2 a.m, he scrawled a note to himself. The idea was simple. If you ligated the pancreatic duct and waited for the digestive tissue to degenerate, you could then extract the substance from the remaining islets without the digestive enzymes destroying it. Banting took this idea to J.J.R. MacLeod, a professor of physiology at the University of Toronto and an established authority on carbohydrate metabolism. MacLeod was skeptical. Banting had no research experience, and the idea, while logical, was not entirely original. But he gave Banting lab space, some dogs, and an assistant, a young medical student named Charles Best, who was just 21 years old. MacLeod then left for a summer vacation in Scotland. What happened over that summer of 1921 is one of the most consequential experiments in medical history. Banting and Best worked through brutal Toronto heat in a cramped, poorly equipped lab. They ligated the pancreatic ducts of dogs, waited weeks for the tissue to atrophy, then prepared extracts from the shriveled pancreases. They injected these extracts into dogs that had been made diabetic by having their own pancreases removed, and the blood sugar dropped. Not perfectly, not cleanly. The early extracts were crude and caused fevers and abscesses at the injection sites. But the blood sugar dropped, and the dogs that had been wasting away started to recover. When MacLeod returned from Scotland and saw the results, he recognized the significance immediately and threw the full resources of his department behind the project. He brought in a skilled biochemist named James Collip, who worked to purify the extract, to remove the contaminants that caused those dangerous side effects. Collip succeeded, producing a preparation clean enough for human use, and in January of 1922, they injected it into a 14-year-old boy named Leonard Thompson at Toronto General Hospital. Thompson was dying. He weighed only 65 pounds. The first injection, using a less purified extract, caused a severe allergic reaction. But 12 days later, they tried again with Collip's improved preparation. Thompson's blood sugar dropped from over 500 milligrams per deciliter to about 120. The ketones in his urine disappeared. He brightened. He gained weight. He lived another 13 years. They called the substance insulin, from the Latin insula, meaning island, a reference to the islets of Langerhans, where it was produced. And the impact was immediate and staggering. Before insulin, a diagnosis of type 1 diabetes was a death sentence, usually within months. Children wasted away in hospital wards, and there was nothing anyone could do. Within a year of insulin's isolation, pharmaceutical companies were producing it at scale, and patients who had been on the verge of death were walking out of hospitals. The Nobel Prize in Physiology or Medicine was awarded to Banting and MacLeod in 1923, just 2 years after the initial experiments, which was extraordinarily fast. But the story has its tensions. Banting was furious that MacLeod shared the prize, feeling that MacLeod had contributed little beyond lab space and had swooped in to claim credit. He split his share of the prize money with Best. MacLeod split his with Collip. The bitterness lingered for decades, and historians still debate the relative contributions. What's clear is that it took all of them: Banting's stubborn idea, Best's tireless lab work, MacLeod's scientific credibility and resources, and Collip's biochemical skill. So how does insulin actually work? Here's where the engineering of blood sugar regulation becomes really beautiful. After you eat a meal, your digestive system breaks down carbohydrates into glucose, which enters your bloodstream. Your blood sugar rises. The beta cells in the islets of Langerhans in your pancreas detect this rise directly. They're essentially glucose sensors, and they respond by secreting insulin into the blood. Insulin then travels throughout your body and acts like a key. It binds to receptors on the surface of your cells, particularly muscle cells and fat cells, and this binding triggers those cells to open glucose transporters in their membranes, molecular doorways that allow glucose to flow from the blood into the cell, where it can be used for energy or stored. Your liver responds to insulin too, absorbing glucose and converting it into glycogen, a compact storage form, like packing away energy for later. As glucose leaves the blood and enters cells, your blood sugar drops. And as it drops, the beta cells sense the decline and reduce their insulin output. Negative feedback again. The same pattern we've been seeing everywhere. But there's a partner hormone that handles the other side of the equation. When your blood sugar drops too low, say you haven't eaten for a while, or you've been exercising hard, the alpha cells of the islets of Langerhans secrete a different hormone called glucagon. Glucagon does roughly the opposite of insulin. It signals the liver to break down its glycogen stores back into glucose and release it into the blood. It can even stimulate the liver to manufacture new glucose from amino acids and other precursors, a process called gluconeogenesis. So you have this beautiful push-pull system. Insulin brings blood sugar down, glucagon brings it up. And between them, they keep glucose in that narrow, safe range. Now, when this system breaks down, you get diabetes. Type 1 diabetes, which is what Leonard Thompson had, is an autoimmune disease. The body's own immune system mistakenly destroys the beta cells, eliminating the source of insulin. Without insulin, cells can't take up glucose, so it accumulates in the blood to dangerous levels while the cells themselves are starving. Type 2 diabetes, which is far more common and accounts for about 90% of cases worldwide, is different. The beta cells still produce insulin, at least initially, but the body's cells become resistant to it. The key doesn't fit the lock as well anymore. The pancreas compensates by producing more and more insulin, but eventually it can't keep up, and blood sugar rises. Type 2 diabetes is strongly associated with obesity, physical inactivity, and genetic predisposition, and it has become one of the defining health challenges of the 21st century, affecting hundreds of millions of people globally. And what's fascinating from an engineering perspective is that the failure mode of type 2 diabetes is essentially a feedback loop that has been overwhelmed. The system was designed to handle a certain range of inputs, the kinds of meals and activity levels that humans experienced for most of evolutionary history. But modern diets, rich in refined sugars and processed carbohydrates, push the system harder and more frequently than it was built to handle. It's like running a machine past its design specifications for years on end. Eventually, components start to wear out and tolerances start to slip. But blood sugar is just one of the variables your body regulates this way. There's another system that operates on a completely different timescale, one that doesn't just respond to moment-by-moment changes, but orchestrates slow, sweeping rhythms that unfold over hours and days, governing when you feel alert, when you feel hungry, when you feel sleepy. And the conductor of this orchestra sits in that same remarkable region we mentioned earlier, the hypothalamus, working in concert with a small, pine cone-shaped gland nestled deep in the center of your brain, a gland that René Descartes once believed was the seat of the soul. It's called the pineal gland, and its primary product is a molecule you've almost certainly heard of, one that is intimately connected to what you're doing right now, lying here in the dark, drifting toward sleep. That molecule is melatonin, and the system that governs its release is tied to something called your circadian clock, a built-in timekeeping mechanism that is honestly one of the most elegant pieces of biological engineering in your entire body. So let's talk about it. Because the circadian system isn't just about sleep. It touches nearly everything. Your body temperature, your hormone levels, your blood pressure, your digestion, your mood, your immune function, even how well you respond to medications. And the story of how we figured it out stretches back centuries to a French astronomer, a bunch of plants, and a very dark closet. The year is 1729, and Jean-Jacques d'Ortus de Méran is curious about something. He's been watching a mimosa plant, the kind whose leaves open during the day and fold closed at night, almost like the plant is going to sleep. And de Méran wonders, is the plant just responding to sunlight, opening when the light hits it and closing when it goes away? Or is something else going on? So he does the obvious experiment. He puts the plant in a dark closet, completely sealed off from any light, and he watches what happens. And here's the thing. The leaves keep opening and closing on roughly the same schedule, even in total darkness. The plant has an internal clock. It knows what time it is without any external cue. Now, de Meuron didn't fully understand what he'd found. It would take more than 200 years for scientists to really work out the mechanism. But that simple experiment established something profound. Living organisms carry time inside them. They're not just passively reacting to the world, they're anticipating it. And that distinction matters enormously, because anticipation lets you prepare. If your body knows that dawn is coming, it can start ramping up cortisol and raising your body temperature before you even open your eyes, so you wake up ready to move rather than spending the first hour of every day scrambling to adjust. In your brain, the master clock sits in a tiny cluster of about 20,000 neurons called the suprachiasmatic nucleus, the SCN for short. It's located right above the optic chiasm, which is the spot where your optic nerves cross on their way from your eyes to the back of your brain. And that location is not a coincidence. The SCN needs light information to stay synchronized with the outside world, and it gets that information through a dedicated pathway from special cells in your retinas. Not the rods and cones you use for vision, but a separate population of cells called intrinsically photosensitive retinal ganglion cells. These cells contain a light-sensitive pigment called melanopsin, and they're particularly responsive to blue light, which is the dominant wavelength of daylight. So even people who are completely blind in the conventional sense, who can't see shapes or colors at all, can sometimes still have functioning circadian rhythms, because these melanopsin cells are doing their own independent light detection and sending timing signals straight to the SCN. Here's what's really clever about the molecular clock itself. Inside each of those 20,000 SCN neurons, there's a feedback loop. And yes, another feedback loop. Because at this point, you're probably noticing that your body runs on feedback loops the way a city runs on electricity. But this one operates on a very specific timescale. It's built from a set of genes. The main ones are called clock and BMAL1 that produce proteins, and those proteins gradually accumulate in the cell over the course of hours. When enough of the proteins build up, they actually go back and suppress the very genes that made them. So the genes turn on, make proteins. The proteins pile up, eventually shut the genes off, and then the proteins slowly degrade, which releases the suppression, and the genes turn on again. One full cycle of this process takes roughly 24 hours. That's where the word circadian comes from, circa meaning about, and diem meaning day. About a day. And I say about because the natural period of the human circadian clock isn't exactly 24 hours. It's closer to 24 hours and 10 or 15 minutes on average, which means that without any external time cues, your internal clock would slowly drift. You'd go to bed a little later each night, wake up a little later each morning, gradually sliding out of sync with the actual rotation of the Earth. This was demonstrated beautifully in experiments conducted in the 1960s and 70s by researchers like Jürgen Aschoff in Germany, who had volunteers live in underground bunkers completely isolated from daylight, clocks, and social cues for weeks at a time. And sure enough, their sleep-wake cycles drifted, settling into rhythms that were slightly longer than 24 hours. Some people drifted to 25-hour days. A few even experienced a phenomenon where their sleep cycle and their body temperature cycle decoupled from each other entirely, running on different periods, which is a strange and disorienting experience from what the volunteers reported. So the SCN needs to be reset every day to stay locked to the actual light-dark cycle, and that resetting process is called entrainment. Morning light is the most powerful entraining signal. When blue-rich light hits those melanopsin cells in your retinas first thing in the morning, it sends a signal to the SCN that says, essentially, it's dawn, reset the clock. And the SCN, in turn, coordinates timing signals throughout your entire body. Because here's the thing that surprised researchers when they discovered it in the late 1990s. Almost every cell in your body has its own clock. Your liver cells have clock genes. Your heart cells have them. Your skin cells, your immune cells, your gut lining. They all run their own local circadian rhythms. But they need the SCN to keep them all synchronized, like an orchestra conductor keeping dozens of musicians on the same beat. Without that central coordination, the peripheral clocks drift apart, and things start to go wrong. This is actually one of the reasons shift work is so hard on the body. If you're working nights and sleeping during the day, your SCN is getting light signals that say one thing, but your eating schedule and social schedule say another, and your various organ clocks end up out of sync with each other. It's not just that you're tired. It's that your liver thinks it's daytime, while your brain thinks it's nighttime, and your digestive system is getting food when it's expecting rest. Long-term shift work has been associated with increased rates of cardiovascular disease, metabolic disorders, and even certain cancers. And while the mechanisms are still being studied, circadian disruption appears to be a significant contributing factor. Now, melatonin fits into this picture as the SCN's chemical messenger of darkness. Here's how it works. During the day, the SCN actively suppresses melatonin production by the pineal gland. But as evening comes and light levels drop, the SCN releases that suppression, and the pineal gland begins synthesizing melatonin from serotonin. Yes, the same serotonin that's involved in mood regulation, which is one reason seasonal changes in daylight can affect how you feel emotionally. Melatonin levels rise through the evening, peak in the middle of the night, usually somewhere between 2 and 4 in the morning, and then decline toward dawn. And melatonin doesn't actually knock you out the way a sleeping pill does. What it does is more subtle. It signals to your body that it's nighttime. It lowers your core body temperature slightly, which promotes sleep onset. It adjusts the timing of other physiological processes. Think of it less as an off switch and more as a dimmer that slowly turns down the lights across your whole system. And that brings us to something worth lingering on: the relationship between your circadian system and your sleep drive. Because they're actually two separate systems that work together, but operate on completely different principles. The circadian system we've been talking about is your internal clock. It promotes wakefulness during the day and allows sleep at night on a roughly 24-hour cycle, regardless of how much sleep you've actually gotten. But there's a second system called the homeostatic sleep drive, and this one is much simpler. The longer you've been awake, the stronger your urge to sleep becomes. It's like a pressure that builds hour by hour, and it's thought to be mediated, at least in part, by a molecule called adenosine. Adenosine is a byproduct of cellular energy use. As your neurons burn through ATP, adenosine triphosphate, the energy currency of your cells, adenosine accumulates in your brain, and adenosine binds to receptors that gradually inhibit the neural circuits that promote wakefulness. So the longer you're awake and the more energy your brain uses, the more adenosine builds up, and the sleepier you feel. When you finally sleep, your brain clears the adenosine, and the pressure dissipates. And here's a fun connection. Caffeine works by blocking adenosine receptors. It doesn't actually reduce the adenosine in your brain. It just prevents the adenosine from binding to its receptors and doing its job. The sleepiness is still there, building up behind the blockade. which is why when caffeine wears off, you can feel suddenly and overwhelmingly tired. All that accumulated adenosine hits its receptors at once. So you have these two systems, the circadian clock saying it's nighttime, sleep is appropriate now, and the homeostatic drive saying, you've been awake for 16 hours, you need to sleep. And normally, they're aligned. They reinforce each other. But when they get out of sync, jet lag is the classic example. You feel terrible, because one system is saying sleep while the other is saying wake, and your body doesn't know what to do. What actually happens in your brain when you fall asleep, though? That's a question that puzzled scientists for a very long time. For most of history, sleep was assumed to be a passive state, a kind of shutting down, like a factory going dark for the night. But that assumption was demolished in 1953, when two researchers at the University of Chicago, Eugene Oserinsky, a graduate student, and Nathaniel Kleitman, his advisor, made a discovery that changed everything. Oserinsky was monitoring the brainwaves and eye movements of sleeping subjects, including his own 8-year-old son, and he noticed something unexpected. At certain points during the night, the sleepers' eyes began moving rapidly beneath their closed eyelids, and their brainwave patterns shifted dramatically. Instead of the slow, rolling waves characteristic of deep sleep, the EEG showed fast, low-amplitude activity that looked almost identical to the waking brain. The sleepers appeared to be deeply asleep, and yet their brains were intensely active. Oserinsky and Kleitman called this rapid eye movement sleep, REM sleep. And when they woke subjects during these periods, the subjects almost always reported vivid dreams. That discovery opened up an entirely new understanding of sleep as an active, structured process with distinct stages that cycle throughout the night. And the architecture of those cycles, the way your brain moves through them in a precise and repeating pattern, reveals something about what sleep is actually for, which turns out to be not one thing but many things. A whole suite of maintenance operations that your body and brain perform while you're lying here, quietly, in the dark. And the list is longer than anyone expected. Let's walk through what a night of sleep actually looks like from the inside. When you first close your eyes and start to drift off, your brain doesn't just plunge into deep sleep all at once. It eases in, like wading into water. The first stage, what sleep researchers call N1, or stage 1 non-REM sleep, is that drowsy, hypnagogic borderland where you're not quite awake and not quite asleep. Your muscle tone starts to relax. Your brain waves, which during wakefulness are fast and desynchronized, lots of different regions firing at their own pace, begin to slow down and synchronize just a little. This stage lasts maybe 5 to 10 minutes, and it's fragile. A door closing, someone saying your name, and you're back. You might not even realize you were asleep at all. But if nothing disturbs you, your brain slides into stage 2, N2, and this is where things start to get interesting. Your brain waves slow further, and two distinctive electrical signatures appear. The first is called a sleep spindle, a brief burst of rhythmic activity, maybe half a second to 2 seconds long, generated by a loop between your thalamus and your cortex. The thalamus, that relay station sitting deep in the middle of your brain, essentially starts gating incoming sensory information. It's closing the doors. Those sleep spindles are part of how your brain walls itself off from the outside world so it can do internal work undisturbed. The second signature is something called a K-complex, a sharp, high-amplitude wave that seems to serve as a kind of gatekeeper, responding to external stimuli and deciding whether they're worth waking you up for. Together, spindles and K-complexes are the sentries of stage 2 sleep, and you spend more time in this stage than any other across the whole night, roughly half your total sleep time. Then comes the deep stuff. Stages 3 and 4, now usually lumped together as N3 or slow wave sleep, are when your brain produces those big rolling delta waves, slow oscillations where vast populations of neurons fire together in synchronized pulses, almost like the whole cortex is breathing in unison. This is the hardest sleep to wake from. If someone shakes you awake during slow wave sleep, you feel groggy, confused, disoriented, that heavy, almost drugged feeling called sleep inertia. And here's the thing about slow wave sleep. It's not just deep rest, it's when some of the most critical maintenance happens. One of the most remarkable discoveries of the last couple of decades involves something called the glymphatic system. And this is genuinely one of those findings that makes you rethink what sleep is fundamentally for. In 2012, a team led by Maiken Nedergaard at the University of Rochester published research showing that during sleep, particularly during slow-wave sleep, the spaces between brain cells actually expand by as much as 60%. Cerebrospinal fluid, that clear liquid that bathes your brain and spinal cord, rushes through these widened channels and essentially flushes out metabolic waste products that have accumulated during the day. One of the key waste products it clears is beta amyloid, a protein fragment that, when it builds up, forms the plaques associated with Alzheimer's disease. Think about that for a second. Your brain has a cleaning system, a literal waste removal infrastructure, and it runs primarily while you sleep. Nedergaard named it the glymphatic system because it functions like the lymphatic system that drains waste from the rest of your body, but it's managed by glial cells, those support cells in the brain that outnumber neurons and do far more than anyone used to think. The implications are staggering. Chronic sleep deprivation doesn't just make you tired and foggy. It may literally allow neurotoxic waste to accumulate in your brain over years and decades, potentially contributing to neurodegeneration. It's one of the most compelling arguments for why sleep isn't optional, isn't a luxury, isn't something you can hack your way out of. Your brain needs that nightly wash cycle. But waste clearance is only one item on the maintenance schedule. Slow-wave sleep is also when your body releases the largest pulses of growth hormone from the pituitary gland. In children and adolescents, this is crucial for physical development. There's real truth behind the old saying that kids grow in their sleep. In adults, growth hormone drives tissue repair, muscle, recovery, and immune function. Your immune system, actually, has a fascinating relationship with sleep. Certain cytokines, signaling molecules that coordinate immune responses, are produced and released in greater quantities during sleep. Studies have shown that people who sleep fewer than 6 hours a night in the week before receiving a flu vaccine produce less than half the antibody response compared to people sleeping 7 or more hours. Your immune system literally learns better when you're well rested. And anyone who's been sick knows the overwhelming urge to sleep that comes with infection. That's not weakness. That's your body deliberately redirecting resources, ramping up immune activity, raising body temperature, all of which work more effectively during sleep. Now, after about 60 to 90 minutes of cycling through these non-REM stages, something shifts. Your brain reverses course, climbing back up through lighter sleep, and then, here it comes, you enter REM sleep for the first time. And REM is strange. Your brain becomes almost as electrically active as it is when you're awake. Your eyes dart back and forth under your lids. Your breathing becomes irregular. Your heart rate fluctuates. and yet your body is essentially paralyzed, a phenomenon called atonia, where your brainstem actively inhibits signals to your voluntary muscles so you don't act out whatever your brain is generating. Because during REM, your brain is generating a lot. This is the primary stage of vivid dreaming, though dreams can occur in other stages too. The paralysis is a safety mechanism, and when it fails, a condition called REM sleep behavior disorder, people can thrash, kick, punch, even get out of bed while dreaming, sometimes injuring themselves or their bed partners. It's more common in older adults and, interestingly, it's now recognized as an early marker for certain neurodegenerative diseases like Parkinson's. But here's what's really clever about the architecture of the night. These stages don't just cycle once. They repeat in roughly 90-minute cycles, 4 to 6 times per night, and the composition of each cycle changes as the night progresses. In the first half of the night, your cycles are dominated by slow-wave sleep, that deep, restorative, waste-clearing, growth hormone-releasing phase. But as the night goes on, the slow-wave portions shrink, and the REM portions grow longer. By the last cycle or two before you wake up, you might be spending 30 or 40 minutes in REM. So the first half of the night is weighted toward physical restoration and metabolic housekeeping, while the second half is weighted toward whatever REM is doing. And what is REM doing? This is where it gets fascinating, and honestly, where the science is still catching up. One of the best supported functions of REM sleep involves emotional processing. Matthew Walker, the neuroscientist at UC Berkeley, who's done so much to bring sleep science to public attention, has described REM sleep as a kind of overnight therapy. During REM, your brain replays emotionally charged experiences from the day, but it does so in a neurochemical environment that's fundamentally different from waking life, specifically norepinephrine, the brain's version of adrenaline, is almost completely absent during REM, so your brain can revisit difficult, stressful, even traumatic experiences without the emotional sting that accompanied them originally. It's processing the memory while stripping away the sharp edge of the emotion. Walker's research has shown that people who get adequate REM sleep show better emotional regulation the next day. They're less reactive to negative stimuli, better at reading facial expressions, more capable of distinguishing friend from foe. People deprived of REM sleep, on the other hand, tend to become emotionally volatile, interpreting neutral faces as threatening, overreacting to minor provocations. And this connects to something clinically important. Many people with PTSD have disrupted REM sleep, and some researchers believe that the failure to properly process traumatic memories during REM is part of what keeps those memories so raw and intrusive. Memory consolidation is the other major function, and it happens across both non-REM and REM in complementary ways. During slow-wave sleep, your hippocampus, that seahorse-shaped structure deep in your temporal lobe that acts as a kind of temporary memory buffer, replays the day's experiences and begins transferring them to the neocortex for long-term storage. Those sleep spindles in stage 2 appear to be involved in this transfer process, acting almost like file transfer protocols between brain regions. During REM, the consolidated memories seem to be integrated with your existing knowledge, creative connections are forged, and insights emerge. There's a famous study where subjects were given a math problem with a hidden shortcut, a pattern in the numbers that, if you noticed it, would let you solve the problem much faster. Subjects who slept on it were significantly more likely to discover the shortcut than those who stayed awake for the same amount of time. And it was specifically REM sleep that predicted the insight. Your brain, during REM, is essentially running simulations, testing combinations, finding patterns that your waking mind was too busy or too constrained to see. So sleep isn't one thing. It's a carefully orchestrated sequence of distinct physiological states, each performing different maintenance operations, timed and ordered across the night in a way that reflects millions of years of evolutionary optimization. And when you cut sleep short, when you set that alarm for 5 hours instead of 7 or 8, you're not just getting less sleep, you're disproportionately losing REM sleep because those long REM periods are concentrated in the final cycles. You're cutting off the emotional processing, the creative integration, the memory consolidation that your brain was saving for last. Which brings us to something even more fundamental than sleep. Something that's been quietly running in the background of everything we've talked about tonight. From your beating heart, to your filtering kidneys, to your cycling brain waves. And that's the nervous system itself, the vast electrical network that coordinates all of it. Because here's the thing about your body. Every system we've discussed, every feedback loop, every homeostatic adjustment, every hormone released on schedule, all of it depends on signals. Signals moving through wires. And those wires, those neurons, operate on principles that scientists spent centuries trying to understand. And the story of how we figured out what a nerve signal actually is, how electricity moves through living tissue, is one of the most fascinating detective stories in all of science. It starts, as so many things do, with an accidental observation. In the late 1780s, an Italian physician named Luigi Galvani was dissecting frogs in his laboratory at the University of Bologna. He had the frog legs pinned to a table, and nearby, one of his assistants was working with a static electricity machine, one of those devices that generates sparks. And Galvani noticed something strange. Every time a spark jumped from the machine, the frog's legs twitched. Even though nothing was touching them, the legs just kicked, as if the frog were still alive. Now Galvani was captivated by this. He spent years experimenting, and he eventually concluded that animal tissue contained its own form of electricity. He called it animal electricity. He believed the muscles themselves stored electrical charge, like tiny biological Leyden jars, and that the nerves served as conductors to release it. His rival, Alessandro Volta, disagreed. Volta thought the electricity was coming from the contact between different metals, the brass hooks and iron railing that Galvani used to hang his frog legs. Volta was so convinced that the electricity was a chemical phenomenon of the metals, not the animal, that he went on to build the voltaic pile, the first true battery, specifically to prove Galvani wrong. And here's the funny thing. They were both partially right. Volta was correct that dissimilar metals in contact with a moist conductor generate electricity, and that insight gave us the battery, which changed the world. But Galvani was also correct that animal tissue generates its own electrical signals. He just didn't have the tools to prove it cleanly. It would take decades before anyone could measure what was actually happening inside a nerve. The real breakthrough came in the 1840s and 1850s, when a German physiologist named Émile Dubois-Reymond developed sensitive galvanometers, instruments that could detect tiny electrical currents, and used them to show that nerves and muscles do indeed produce measurable electrical signals. He demonstrated that when a nerve fires, there's a detectable change in electrical potential that travels along its length. He called it the negative variation, and it was the first direct measurement of what we now call the action potential. His student, Hermann von Helmholtz, went even further. Helmholtz actually measured the speed of the nerve impulse in a frog's leg. People had assumed nerve signals traveled at the speed of electricity, essentially instantaneously, like current through a wire. But Helmholtz showed it was much slower, about 27 meters per second in a frog nerve. That's fast, but it's nowhere near the speed of light, which meant the nerve signal wasn't just raw electricity flowing through a conductor. Something more complex was happening. And this is where it gets really beautiful, because what's actually happening inside a neuron is an elegant electrochemical process that took until the mid-20th century to fully work out. The heroes of that story are Alan Hodgkin and Andrew Huxley, two British physiologists working at Cambridge, who in the late 1940s and early 1950s performed a series of experiments on the giant axon of the squid, a nerve fiber so large you can actually see it with the naked eye, about 1 millimeter in diameter, which made it possible to insert electrodes directly inside the nerve cell for the first time. Here's what they found, and here's what's happening right now in every nerve in your body as you lie there. A neuron at rest isn't electrically neutral. There's a voltage difference across its membrane. The inside of the cell is about 70 millivolts more negative than the outside. That's called the resting membrane potential. It exists because the cell membrane is studded with tiny protein pumps, sodium-potassium pumps, that constantly push 3 sodium ions out of the cell for every 2 potassium ions they pull in. Since sodium and potassium are both positively charged, but you're exporting more positive charges than you're importing, the inside ends up slightly negative. Think of it like a battery that's always being charged. The cell is spending energy, burning ATP, to maintain this electrical imbalance, this state of readiness, every second of every day. Now, when a signal arrives, when something stimulates the neuron enough to push that voltage past a threshold, roughly negative 55 millivolts, something dramatic happens. Voltage-gated sodium channels in the membrane snap open. These are protein channels that literally change shape when they sense a voltage change, creating a pore that allows sodium ions to flood into the cell. And sodium rushes in fast because it's being pulled by 2 forces at once. The concentration gradient, since there's way more sodium outside than inside, and the electrical gradient, since the inside is negative and sodium is positive. So sodium pours in, and the voltage at that spot on the membrane swings wildly positive, up to about plus 30 or 40 millivolts. This is the action potential. This is the nerve impulse. But it lasts only about a millisecond, because almost immediately, the sodium channels inactivate. They have a built-in timer, essentially a little molecular gate that swings shut. And voltage-gated potassium channels open, allowing potassium to rush out of the cell, which brings the voltage back down. Actually, it overshoots slightly, going a bit more negative than resting potential for a brief moment before the pumps restore everything to baseline. The whole event takes maybe 1 to 2 milliseconds at any given point along the nerve. And here's what's clever. That spike of positive voltage at one spot on the membrane is enough to trigger the voltage-gated sodium channels in the adjacent patch of membrane to open, which triggers the next patch, and the next, like a line of dominoes falling. The signal propagates down the length of the axon, not because electricity is flowing through it like current through a copper wire, but because each segment of membrane is regenerating the signal fresh. It's more like a stadium wave. Each person stands up because the person next to them stood up, and the wave travels around the stadium, even though no individual person is moving around the stadium. This is why Helmholtz measured a speed of meters per second rather than the speed of light. The signal has to be chemically regenerated at each point along the way. Now, 27 meters per second is fine for a frog, but your body is bigger, and some signals need to travel faster. Your motor neurons, the ones that control your muscles, need to send signals from your spinal cord to your toes. And if those signals traveled at frog speed, your reaction times would be terrible. So evolution came up with an ingenious solution: myelin. Certain glial cells, Schwann cells in the peripheral nervous system, oligodendrocytes in the brain and spinal cord, wrap themselves around axons in tight spirals, creating a fatty, insulating sheath. Myelin acts like the rubber insulation around an electrical wire. It prevents ion leakage through the membrane, which means the electrical signal can jump from one gap in the myelin to the next. These gaps are called nodes of Ranvier, and they're packed with sodium channels. The signal essentially leaps from node to node, a process called saltatory conduction, from the Latin word saltare, to jump, and this speeds things up enormously. A myelinated human motor neuron can conduct signals at about 120 meters per second. That's over 260 miles per hour. Fast enough that the delay between your brain deciding to move your hand and the signal reaching your fingers is only a few milliseconds. And when myelin breaks down, as it does in diseases like multiple sclerosis, you can see exactly how critical it is. Signals slow down, become unreliable, arrive garbled. Muscles weaken, coordination falters, vision blurs. The wires are still there, but the insulation is damaged, and the system can't function properly. It's one of the clearest examples in medicine of how structure and function are inseparable. So that's the signal traveling down a single neuron. But here's the next question. How does the signal get from one neuron to the next? Because neurons don't actually touch each other. There's a tiny gap between them, about 20 nanometers wide, called the synapse. And crossing that gap requires a completely different mechanism. When the action potential reaches the end of an axon, the axon terminal, it triggers voltage-gated calcium channels to open. Calcium flows in, and that calcium signal causes tiny vesicles, little membrane-bound bubbles filled with chemical messengers called neurotransmitters, to fuse with the cell membrane and release their contents into the synaptic gap. Those neurotransmitter molecules drift across the gap. It only takes a fraction of a millisecond. The distance is so small, and bind to receptor proteins on the next neuron's membrane. Depending on which neurotransmitter it is and which receptor it binds, this can either excite the next neuron, pushing it toward firing its own action potential, or inhibit it, making it less likely to fire. And this is where the real computational power of the nervous system lives, because a single neuron in your brain might receive input from 10,000 other neurons simultaneously. Some of those inputs are excitatory, some are inhibitory, and the neuron is essentially summing them all up, moment by moment, deciding whether the total input crosses the threshold for firing. It's not a simple relay. It's an integration device, and with roughly 86 billion neurons in the human brain, each with thousands of synaptic connections, the number of possible states, the sheer combinatorial complexity, is staggering. It dwarfs anything we've ever built. The neurotransmitters themselves are fascinating molecules, and you've probably heard of the major ones even if you didn't know exactly what they do. Acetylcholine was the first one discovered by Otto Loewi in 1921 through a beautifully simple experiment involving 2 frog hearts in a shared bath of saline solution. He stimulated the vagus nerve of one heart, which slowed it down, and then showed that the saline from that heart, transferred to the second heart, also slowed the second heart, proving that the nerve released a chemical substance, not just an electrical signal. He called it Vagusstoff, vagus substance, and it turned out to be acetylcholine. Lowy later said the idea for the experiment came to him in a dream. He woke up, scribbled it down, went back to sleep, and in the morning couldn't read his own handwriting. The next night, the dream came again, and this time he got up immediately and went to the lab. Science sometimes literally happens in your sleep. Then there's dopamine, which you've probably heard called the reward chemical, though that's an oversimplification. It's really more about motivation and prediction, about signaling that something is worth pursuing. There's serotonin, deeply involved in mood regulation, gut function, and sleep-wake cycles. And remember, most of your body's serotonin is actually produced in your gut, not your brain. There's norepinephrine, which we talked about earlier in the context of REM sleep, the alertness and stress response chemical that gets deliberately shut off during dreaming. There's GABA, gamma-aminobutyric acid, the brain's primary inhibitory neurotransmitter, the one that calms neural activity down, and the one that drugs like benzodiazepines and alcohol enhance, which is why they're sedating. And there's glutamate, the primary excitatory neurotransmitter, the gas pedal to GABA's brake. The balance between excitation and inhibition in the brain is extraordinarily precise, and when it's disrupted, the consequences are immediate and dramatic. Too much excitation without enough inhibition, and neurons fire uncontrollably. That's essentially what happens during an epileptic seizure. Too much inhibition, and consciousness itself dims. Your brain, right now, is maintaining a delicate equilibrium between these forces, and the fact that it does so reliably, minute after minute, year after year, is one of the most remarkable feats of biological engineering there is. But the nervous system isn't just the brain. There's an entire division of it that you almost never think about, that operates almost entirely below conscious awareness. And it's been managing your body all night while we've been talking. It's called the autonomic nervous system, and it controls your heart rate, your breathing, your digestion, your blood pressure, your pupil dilation, all the things that keep you alive without you having to remember to do them. And it has two branches that work in opposition, like a thermostat with both a heater and an air conditioner. The sympathetic branch is your accelerator. It speeds up the heart, dilates the airways, diverts blood to muscles, releases adrenaline. It's the fight-or-flight system. The parasympathetic branch is your brake. It slows the heart, promotes digestion, conserves energy. It's sometimes called the rest and digest system. And right now, as you lie here in the dark, your parasympathetic system is gently dominant. Your heart rate is settling. Your digestion is quietly working. Your breathing is slow and deep. Your body is in maintenance mode, and the autonomic nervous system is the foreman overseeing the whole operation. What's remarkable is how this system connects to everything we've already talked about. Remember the baroreceptors we mentioned? The pressure sensors in your arteries that help regulate blood pressure? Those signals travel through autonomic nerves. The way your kidneys adjust filtration rate, partly regulated by sympathetic nerve input. The circadian signals from the SCN that tell your pineal gland to release melatonin. They travel through a surprisingly long sympathetic pathway that runs up through the spinal cord and loops through a ganglion in your neck before reaching the pineal gland deep in your brain. Everything is connected through this network, and the nervous system is the thread that ties it all together. And there's another nervous system you might not have heard of, one that scientists have only recently started to appreciate fully. It's called the enteric nervous system, and it lives in the walls of your gastrointestinal tract. It contains somewhere around 500 million neurons, more than your spinal cord, and it can operate largely independently of the brain. It manages the complex choreography of digestion, the rhythmic contractions that move food along, the secretion of enzymes at the right time, the regulation of blood flow to absorb nutrients. It's sometimes called the second brain, and while that's a bit of an exaggeration, the fact that your gut can coordinate such complex behavior on its own, with its own sensory neurons, motor neurons, and interneurons, tells you something profound about how distributed the intelligence in your body really is. Your brain isn't a central computer running everything. It's more like the CEO of a vast organization where regional managers handle most of the day-to-day operations, autonomously, only escalating to headquarters when something unusual happens. Which brings us to the immune system, something we touched on briefly when we talked about sleep, but which deserves a much closer look because it's arguably the most sophisticated defense network ever engineered by evolution. And I don't use that word lightly, engineered, because when you look at what the immune system actually does, the precision of it, the layered redundancy, the ability to learn and remember and adapt, it makes the most sophisticated human-designed defense systems look like a padlock on a screen door. So let's talk about it. Let's talk about what's protecting you right now, tonight, as you lie here breathing in a world absolutely teeming with things that would love to use your body as a warm, nutrient-rich home. The first thing to understand is scale. You're surrounded by microorganisms. They're on your skin, in your mouth, lining your gut, floating in every breath you take. A single gram of soil contains something like 40 million bacteria. A milliliter of seawater holds about 1 million. The air in this room, wherever you are, has thousands of microbes drifting through it. Most of them are harmless. Many are beneficial. Your gut microbiome, for instance, is essential for digestion, vitamin synthesis, and immune development. But some fraction of what you encounter every day would, if given the chance, invade your tissues, hijack your cells, and replicate at your expense. Bacteria, viruses, fungi, parasites. They've been locked in an evolutionary arms race with multicellular organisms for hundreds of millions of years, and your immune system is the accumulated wisdom of that war. The ancient Greeks had some notion that surviving a disease could protect you from getting it again. Thucydides noted during the plague of Athens around 430 BCE that those who recovered were able to nurse the sick without falling ill a second time. But they had no framework for understanding why. For most of human history, disease was attributed to miasma, to imbalanced humors, to divine punishment. The idea that tiny invisible organisms could cause illness, germ theory, didn't gain real traction until the mid-1800s, when Louis Pasteur in France and Robert Koch in Germany independently built the case through meticulous experimentation. Pasteur showed that microorganisms caused fermentation and spoilage, then extended the logic to disease. Koch developed his famous postulates, a set of criteria for proving that a specific microbe causes a specific disease, and used them to identify the bacteria responsible for anthrax and tuberculosis. By the 1880s, the germ theory revolution was transforming medicine. But understanding that germs cause disease is one thing. Understanding how your body fights them is something else entirely, and that story unfolded much more slowly. Let's start with the first line of defense, because it's beautifully simple. Your skin. You're wrapped in a continuous barrier of keratinized epithelial cells. Tough, dead cells packed with a protein called keratin that makes them waterproof and resistant to penetration. It's essentially biological armor. Your skin's surface is slightly acidic, around pH 5.5, which discourages many bacterial species. It's colonized by friendly bacteria that compete with potential invaders for resources. And anywhere there's an opening, your eyes, your nose, your mouth, your respiratory tract, your gut, you have other defenses. Tears contain lysozyme, an enzyme that breaks down bacterial cell walls. Your nasal passages are lined with mucus that traps particles, and tiny hair-like cilia that sweep them back out. Your stomach produces hydrochloric acid strong enough to dissolve metal, which sterilizes most of what you swallow. These barriers are ancient and effective, and most of the time, they're all you need. But sometimes something gets through. A cut in the skin, a pathogen that survives the acid bath, a virus that lands on a mucosal surface and manages to infect a cell. And now the real immune system kicks in. There are two major branches, and here's where it gets really elegant. The first is called the innate immune system, and it's the rapid response force. It's fast, it's non-specific, and it's been around in some form for at least 500 million years. You find versions of it in insects, in sea stars, in organisms that diverged from our lineage long before vertebrates existed. The innate system doesn't need to know exactly what it's fighting. It recognizes broad patterns, molecular signatures that are common to many pathogens, but absent from your own cells. These are called pathogen-associated molecular patterns, or PAMPs, and your innate immune cells have receptors, pattern recognition receptors, that detect them. One of the most important families of these receptors, called toll-like receptors, was first discovered in fruit flies in the 1990s by Jules Hoffman and his colleagues, work that eventually earned a Nobel Prize in 2011. The discovery revealed that even organisms with no adaptive immunity have sophisticated pathogen detection systems. When an innate immune cell, say a macrophage patrolling your tissues, detects a bacterial invader, it does something wonderfully direct. It eats it, literally engulfs it in a process called phagocytosis, pulls it inside, and destroys it with enzymes and reactive oxygen species. This was first observed by Elie Metchnikoff in the 1880s, and the story is wonderful. Metchnikoff was a Russian zoologist studying starfish larvae, transparent little creatures you could watch under a microscope. He poked a rose thorn into a larva and watched as mobile cells gathered around the foreign object and tried to engulf it. He realized he was watching a fundamental defense mechanism, one that didn't require antibodies or anything specific to vertebrates. He called these cells phagocytes, from the Greek for eating cells, and he spent the rest of his career championing the idea that cellular immunity, the body's cells physically attacking invaders, was the primary defense against infection. He shared the 1908 Nobel Prize with Paul Ehrlich, who championed the opposing view that soluble factors in the blood, what we now call antibodies, were more important. Turns out they were both right. The immune system uses both strategies, and they work together in ways neither man could have imagined. So your innate system responds within minutes to hours. Macrophages and neutrophils, the most abundant white blood cells, the ones we talked about earlier, rush to the site of infection. They release chemical signals called cytokines that trigger inflammation. Blood vessels dilate, become more permeable, and more immune cells flood in. That redness, swelling, heat, and pain you feel around a wound, that's inflammation, and it's not a malfunction. It's the immune system working exactly as designed, creating conditions that favor immune cells and hinder pathogens. Fever, too. When your body raises its thermostat set point, remember how we talked about the hypothalamus regulating temperature? Fever is an active immune strategy. Many pathogens replicate optimally at normal body temperature, and even a degree or two of elevation can slow them down while enhancing certain immune functions. But here's the thing about the innate system. It's fast, it's powerful, but it's generic. It can't adapt to a specific threat, can't remember a pathogen it's seen before, can't mount a more targeted response the second time around. For that, you need the adaptive immune system. And this is where the engineering gets truly extraordinary. The adaptive immune system is unique to vertebrates, appearing first in jawed fish around 450 million years ago, and it solves a problem that seems almost impossible. Your body needs to be able to recognize and respond to essentially any molecular shape it might encounter. Any protein fragment from any virus, any surface molecule from any bacterium, including pathogens that don't even exist yet, that might evolve next year or next century. How do you prepare for threats you've never seen? The answer is one of the most beautiful solutions in all of biology. Your body generates diversity randomly, then selects for usefulness. Here's how it works. You have two main types of adaptive immune cells: B cells, which mature in the bone marrow, and T cells, which mature in the thymus, a small organ behind your breastbone that's most active in childhood. Each B cell and each T cell carries a unique receptor on its surface, and that receptor is generated through a process of random gene rearrangement. During development, the gene segments that encode these receptors are shuffled and recombined in essentially random combinations, a process discovered by Susumu Tonegawa in the 1970s, earning him the 1987 Nobel Prize. The result is staggering. Your body produces something on the order of 1 billion different receptor configurations, each one capable of binding to a different molecular shape. Most of these will never encounter their matching antigen, but the ones that do, the ones that happen to fit a fragment of an invading pathogen, those cells get activated. Think about what that means. Your body doesn't need to predict what pathogens you'll encounter. It just generates an enormous random library of possible responses and then lets the pathogen itself select which response gets amplified. It's like printing 1 billion different keys and waiting to see which lock shows up. When a B cell encounters an antigen that fits its receptor, it begins to divide rapidly, producing clones of itself. Some of these clones become plasma cells that secrete antibodies, soluble versions of that receptor, into the blood and tissues, where they bind to the pathogen, neutralize it, tag it for destruction by other immune cells. Other clones become memory B cells that persist for years, sometimes decades, ready to mount a faster, stronger response if the same pathogen appears again. That's immunological memory, and it's the entire basis of vaccination. And vaccination itself, what a story. Edward Jenner in 1796, a country doctor in Gloucestershire, England, who noticed that milkmaids who'd had cowpox seemed immune to smallpox. He took material from a cowpox lesion on a milkmaid's hand and inoculated an 8-year-old boy named James Phipps. Then, weeks later, he deliberately exposed the boy to smallpox. The boy didn't get sick. Jenner had demonstrated that exposure to a related but less dangerous pathogen could confer protection against a deadly one. He called it vaccination, from vacca, the Latin word for cow. He didn't understand the mechanism. B cells, T cells, antibodies, none of that was known. But he'd stumbled onto one of the most powerful principles in medicine. Today, vaccination has eradicated smallpox entirely, nearly eradicated polio, and saves millions of lives every year. It works because of immunological memory, because your adaptive immune system remembers. T cells are the other half of this story, and they're equally remarkable. While B cells handle threats in the blood and extracellular spaces, T cells deal with problems inside your cells, particularly viral infections, where the pathogen hides within your own cells and can't be reached by antibodies. Cytotoxic T cells, also called killer T cells, can recognize when one of your own cells has been infected because infected cells display fragments of the virus on their surface, like a distress flag, and they destroy that cell before the virus can replicate further. Helper T cells coordinate the broader immune response, releasing cytokines that activate B cells, boost macrophage activity, and orchestrate the whole campaign. The loss of helper T cells is exactly why HIV is so devastating. It specifically targets and destroys CD4 positive helper T cells, gradually dismantling the immune system's ability to coordinate responses, leaving the body vulnerable to infections that a healthy immune system would handle easily. And here's something that ties back to everything we've talked about tonight. Your immune system doesn't operate in isolation. It's deeply intertwined with every other system we've discussed. Remember how we talked about sleep and immune function? During deep, slow-wave sleep, your body increases production of certain cytokines and shifts immune activity toward surveillance and repair. The stress hormones regulated by your autonomic nervous system, cortisol, adrenaline, directly suppress certain immune functions, which is why chronic stress makes you more susceptible to illness. Your circadian clocks, those peripheral oscillators we talked about in every tissue, they regulate immune cell trafficking. The number of immune cells circulating in your blood actually varies across the day, peaking in the evening and dipping in the early morning. Even your gut microbiome, managed by that enteric nervous system, plays a crucial role in immune development. Something like 70% of your immune tissue is associated with the gastrointestinal tract. And that's really the deepest lesson here, isn't it? Not any single system, not any one discovery, but the way it all connects. Your heart pumps blood that carries oxygen bound to hemoglobin in red blood cells produced in response to erythropoietin from your kidneys, which also filter that blood and regulate its pH and pressure, while your nervous system coordinates the heart rate and vessel tone through autonomic pathways timed by circadian clocks in the SCN, and your immune cells ride that same bloodstream to patrol tissues maintained by hormones released during sleep stages, orchestrated by adenosine accumulation and circadian melatonin signals. Every system we've talked about tonight is in constant conversation with every other system, through hormones, through nerves, through the shared medium of the blood, through timing signals, through feedback loops within feedback loops. Claude Bernard was right all those years ago. The constancy of the internal environment is the condition for free life. Your body maintains this extraordinary internal stability: temperature, pH, glucose, oxygen, fluid balance, immune surveillance, not through any single master controller, but through a distributed network of systems that monitor, communicate, adjust, and compensate moment by moment, breath by breath, heartbeat by heartbeat. Right now, while you're lying here, not thinking about any of it. And maybe that's the most remarkable thing of all. You don't have to think about it. You don't have to remember to make your heart beat, or tell your kidneys to filter, or instruct your immune cells where to go. Billions of years of evolution have built these systems so robust, so redundant, so elegantly self-regulating that they just work, quietly, in the dark. While you rest, your body is doing all of this right now. Your diaphragm is contracting and relaxing, drawing air in and pushing it out. Your heart is beating somewhere around 60 or 70 times a minute, sending blood on its endless circuit. Your kidneys are filtering, your liver is processing, your immune cells are patrolling, your neurons are firing, maintaining the delicate balance of excitation and inhibition that lets you drift between waking and sleeping. Adenosine is building in your synapses, the sleep pressure we talked about, and melatonin is circulating, and your body is preparing to do what it does so well: repair, consolidate, restore. You don't need to understand all of it. You don't need to remember every detail. The stories are there if you want them. Vesalius and his stolen skeletons, Harvey and his ligatures, Starling and his heart-lung preparation, Banting and Best and their desperate summer with diabetic dogs, Oserinsky watching his son's eyes move in sleep, Hodgkin and Huxley and their squid axon, Lowy dreaming about frog hearts. These are human stories, stories about curiosity and persistence, and the slow, painstaking work of figuring out how we're made. And the beautiful thing is, we're still figuring it out. There's so much left to learn. But for now, you don't need to figure anything out. Everything is taken care of. Your body knows what to do. It's been doing it since before you were born, and it will keep doing it tonight. So let it. Let the systems run. Let the feedback loops close. Let the signals travel their well-worn paths. You are, right now, the most complex and finely tuned system that has ever existed on this planet. And all you have to do is rest. Just breathe and rest, and let your extraordinary body do what it was built to do.